Hi, everybody. Today I'm going to be speaking about some of the technologies that we've developed here at AOML for better monitoring and protecting U.S. coral reefs. And we all know that coral reefs are very important, but they're also quite valuable. They provide numerous ecosystem services through fisheries, tourism, storm protection, and ultimately serving as the single highest concentration of biodiversity in the marine realm, giving billions of dollars to the U.S. economy every year. And as my colleague, Dr. Barbaro, so expertly introduced, atmospheric CO2 is rising. This leads to higher concentrations of carbon dioxide in the water. And this is accompanied by a decline in seawater pH in a process known as ocean acidification. And at AOML, we have shown that ocean acidification has slowed down coral growth, increased erosion. It has directly impacted fish stocks, and it has led to fundamental shifts in ecosystem structure and function of coral reefs. Now, I'm going to be talking about technologies that we've used to characterize organismal responses, to monitor reef conditions, to experimentally test the resilience of corals, and ultimately tools that we are developing to increase the efficacy and efficiency of restoration efforts. We've developed CT scanning technologies that allow us to look inside coral skeletons, to trace back annual density bands to determine how they've been growing through time under different stressors. We've also developed 3D scanning technologies. This allows us to digitally quantify the complex surface morphologies of different corals, and we can do this in a manner that's fast enough that we can do this on live corals in the laboratory. We developed micro CT scanning technologies that allow us to look inside reef framework habitats at a very, very fine scale to understand how quickly they are dissolving away under stressors such as ocean acidification. Now, one of the problems with monitoring ocean acidification, especially in shallow water environments, is the high level of variability, as you can see here from our near real-time CO2 buoy at Chica Rocks in the Florida Keys. This necessitates a large amount of sampling in order to be able to accurately quantify ocean acidification. Ship time, as we know, is expensive, so are instruments, and sometimes it can be extremely dangerous or time-consuming to be in the field, especially during adverse, interesting conditions. So we've developed subsurface automated samplers in-house at AOML that simply put allow us to collect water without divers or assets in the field. This allows us to expand our sampling efforts in both space and time. We've done this from the ground up, designing the circuitry, the software, the hardware, 3D printable CAD models, and providing all of this information to any of our collaborators online, free of charge, in an entirely open source manner. And tons of labs are integrating this into their monitoring design. We've started to find out that variability is likely very important for the actual modulating the response of different organisms to ocean acidification stress. But to test this, we really needed a laboratory that was able to control ocean acidification at a pretty unprecedented level of detail. And so as many of you saw yesterday, we've created the experimental reef lab across the street at CMUS that is a fully automated robotic system where, again, we have designed all of the hardware, all of the software in-house so that we can manipulate not just mean ocean acidification conditions, but the variability around that mean for both pH as well as temperature. And we've used this, and as you can see on the left, we are ramping conditions as well as the how accurate and precise they are over time. So this is something that is actually working quite well, I'm very happy to say. We've used this in several experiments. This is one that we used on endangered Acropora cervicornis, where we found under highly variable contemporary conditions that actually increases the calcification, the growth potential of this species, potentially providing present-day refugia. We've also seen that there's an incredible amount of genetic variability with how these organisms grow and with their response to different stressors. And we think that we can use this to actually inform restoration efforts. But one of the problems is that global coral reef die-offs are such a huge problem that it's important that we understand and keep in mind how we can scale our efforts to combat this issue as we go forward. So one of the things that we're doing right now is developing technologies for fully automated evaluation of the performance of different coral genotypes. So again, that we can scale this evaluation and really affect meaningful change to this global problem. And with that, I'd like to thank a lot of our funding sources and you guys here today.